when I say a bit of history, it's just a bit of history. I think it's clear that the Sanger sequencing was really powerful and was the way for many, many, many years to generate to get the information of a sequence, and even quite long ones. And the first time I saw one of those, I even didn't know what was that. But you get the information about the sequence of RNA. I don't want to make a big emphasis on that. I will explain the figures. So basically, the work on the NGS, the next generation sequencing, I'm going not to talk about all the history, everything. We don't have time for all those things. You have a lot of papers and a lot of information if you want to dig more into that. I kind of specify more into the Illumina pipeline. Sanjay here is the one who is, yes, you don't mind doing that? I think it's good because we have a lot of different expertise, and you are the one of them that has been running the sequencing pipeline in the unit. And he if I say anything that is not completely accurate, just jump in. So basically, I will just kind of show a bit of a snapshot of how this is working, and you have three main steps when we are generating this sequencing data. At the end, what we want is to have we kind of cut the RNA or the DNA into pieces, and we want to have information of all those pieces, right? We want to have the full sequence of them. So, and that's kind of the steps on the process. We cut the DNA or we cut the RNA, and then we add adapters to the extremes. We can add adapters. I will tell later on. We can also add like barcodes or demultiplexing. But these adapters are very useful because you put this into kind of a surface, and they kind of attach to the surface, and they start kind of multiplying. I will go into detail for each one of those steps a bit more. And you create kind of clusters that are having exactly the same sequence of DNA. The idea of creating those clusters is because later on, when you do the identification through the fluorescence, you will have enough signal to recover what nucleotide was there. And this is this stage, at the sequencing part. If we look into a bit more, as I was saying, you get the DNA, you fragment it, you add the adapters, and then you are kind of prepared for going into the sequencing. This is just a very short version. I'm more into the bioinformatics. I think this is what we aim here, but it's good to have this idea. Why? Because when we are doing defragmentation, we have different decisions to make. How long do you make the fragmentation? You can make fragments of 200, 100, 300, but then you make a fragment of 200 and then you add the adapters. That means that you are adding something that is even longer, and this will also maybe affect later when we do the sequencing. The point here is we want to sequence this we call fragment and this fragment. And of course we can kind of use this the technology that we will show later for the fluorescence, but the problem is when you do this with just one single DNA strand, it's not strong enough, the signal that they get. So what they wanted to do is to say, well, around each one of those locations, we are going to create a cluster of DNAs with the same sequence. So they are allowing that's why you have two adapters, because then they bind, they replicate, they go back, they bind, they replicate, and eventually you have set with exactly the same sequence close by, and you are able to kind of sequence them because you get enough signal for that. In the third step, which the idea is that right now you have your sequence here, your fragment, and of course I'm showing one, but it's basically a cluster of them, right? And iteratively you are adding a nucleotide, and this will give like a signal, and this is exactly what you are kind of scanning here. And then you do step by step, and you are having like information for each one of the clusters. Just to make it kind of a bit more clear, imagine this is the slide. Each one of those dots is where the clusters has been generated. So from time to time, we are scanning this figure, and we are getting information about, okay, it's green, so it's T. It's blah, it's a nucleotide. And for each one of them, we need to follow over the time, and this will give us the sequence. This is a very kind of short version explanation. I don't know how many of you has worked with macroarrays, but basically it has similar problems to the macroarrays, even a bit more complex. Because in the macroarrays you also have the fluorescence, you identify the signal of the fluorescence, and the problem of quantifying it makes a challenge. One of the aspects is, right when you have when you go to the machine, you use like flow cells. Flow cells is, I mean, like it has separate 8 or 2 lines depending on it, and in each lane, 8 lanes, and in each lane you are adding your sample. The problem is, if we only put 1 sample per lane, that would be very expensive, because it will give like a lot of reads, but we are not able to do more than 8 samples for every flow cell. That will become extremely expensive. So of course then we need to do something like multiplexing, and what it is basically, in addition to when you prepare your sample, when you prepare your library, in addition to the adapter, you are adding something that is called the barcode. So you add a sequence of nucleotides that will help you to identify what is the origin of the sample. Then you are free to mix them into the same line, because then after that you are able to recover this is this sample and this is this sample based on this barcoding. What is the drawback? The drawback is you need to sequence the barcode. Okay? And sequencing is getting cheaper, but it's an expensive thing. And we will see later what are the kind of quality measures that we can have for the sequence. I mean, we can sequence more, that means we get more reads, more fragments that we are having information, or we can make them longer. What do you think is the benefit of having a fragment to be longer? So imagine we have a sequence, I say a fragment, and we have 20 pair bases. Now, and I will show later, the challenge is these 20 pair bases, which gene they pertain to. Because our aim is to find which gene is originated. If I give you 20 pair bases, how likely it is it can map to the genome? Probably in many locations. If I give you 50, it's less likely to bind to many locations. And if I give you 100, it should go to the original place, if everything will be perfect in an ideal world. That means that by investing into adding more, you are making easier to map to the right original location. If we talk about the DNA assembly, so you can sometimes, if you work in the context of genotypes, you can get DNA information from an individual and try to map to a specific location, try to find if this person had a genotype, a SNP here, etc. But you can try also to recreate the genome of a given person. And for doing that, what you are doing is kind of overlapping many different reads. So if you have 20 pair bases, it's very complex. The longer you have, the better it is to create this kind of framework of continuing reads. So if we are a barcode, which we do in a daily basis, we are using some of the reads that we are getting to identify the sample and not to map it to the genome, which makes it a bit more expensive. So it's always kind of a trade-off, and we need to make decisions. The other thing is the pair-end, single-end. I was showing you, and this is really important. I'm talking most of the time about a fragment, okay? So I get this fragment of DNA, and then I start sequencing in one direction. This is what we call like single end. Sorry, it's a single end. But we also can start sequencing in one direction and try to sequence in the other direction. And maybe we have a hole in the middle that we don't know, but we have a read. We have a read here, and we know that they have to map close by. They have to map to the same chromosome. They cannot be kind of chromosome 1, chromosome 20. This is what we call the parent. This is really, really, really powerful. And of course, more expensive because for each fragment, you are getting 2 reads. So if we have 50 per bases, we get 50 and 50, 100. The price is getting higher. It's not kind of the scope of the course to discuss about what is, I mean, there are many papers talking about parent, single end, multi-sequenceless read. I think in many contexts, parent is very important. In the context of transcriptomics, it's really, really important. We will see later. In the context, for instance, of exons, because it really helps you to identify kind of introns or splice junctions, etc. So now I have one question for you. I was showing the library preparation, cluster amplification, sequencing, and then after sequencing, what we get is, okay, I have this sequence of DNA. Where does it go? And we align and we do the data analysis. My question for you is, where do you think bioinformatics pipeline is starting here? From my point of view, I think the bioinformatics starts really here, because we need to transform fluorescent signals into nucleotides. So I wanted just to put this example, because we need to move from this into this part. I was saying before, when the macroarrays appear, I guess everyone is kind of have read about macroarrays at some point, right? Anyone not? Okay. So you remember it goes through the fluorescence, and then the problem was we need to move from the fluorescence to a given value. So where do we take the fluorescence? And the fluorescence was coming like from square, and sometimes you have like a circle, and you say, okay, where from this image, where I am scanning, or which information of this image I'm using to estimate the strength of the signal. That was kind of that problem. In our problem, is a bit different. It we have to identify the locations where the clusters are coming. Once we have identified a location, we have to follow it over the time. And then we have to say, how much do we trust this is a T? And we will estimate the error of the tree, of this being a T. Comparing to macroarray, which at the beginning there was a lot of people, or I think quite some people working into better algorithms for kind of this scanning. In the Illumina, they say, well, they develop their own kind of packages, originally Cassava, then you have this BCL to FastQ. So it's the base calling to FastQ, it will give us the sequencing. And right now you have, I mean, this is kind of to be discontinued, and this is the type of well, this is one of the softwares that we can use. So there is not that much development, bioinformatically speaking, into that, but this is an important aspect to consider, especially when we think about quality controls and everything. So after that, what I was saying is that now we have a sequence, right? What do we do with the sequence? It actually will depend, or I mean, one thing is what is a general pipeline. It will depend a lot in which data types we are using. So those are the ones that we will talk here for the NGS, and there is no single kind of pipeline. Everyone has like specific things. What I'm going to do now is to, there is different pipelines for each one of those kind of protocols, but there are shared parts. And shared parts that we can think about what is the quality, what is the things, and those shared parts I'm going to kind of discuss before I go into the RNA-seq. So before we go into the FastQ, we need to check about a quality control. Okay, we are getting these images, we are getting the VCL files, and then we move to FastQ. There is quality controls at this level, and Sanjay knows quite well all of them. Like how many clusters you have generated. Because the number of clusters will tell you the number of reads that you are getting. The second point will be, okay, you have 1 million clusters, fine. But now, instead of having one sample, I'm using 3. That means that I have 3 barcodes. How many of those reads are getting into any one of those samples? Because some of them maybe are not filtered into any one. And why is that? Because you have problems maybe in recognizing it. You have maybe problems with the chemistry and something has happened there. So you have the percentage of the fragments that has been really identified to the original to the original sample. Those kind of things are to be considered here. There are other ones. I will not talk about them, but some of them are also kind of taken when we talk about the FastQ files. So if most of the cases, we don't work with a machine in our lab. At least for us in the beginning, we didn't have the sequencing machine, so we have to go to facilities, right? And in many cases, the facilities are making fantastic job, but they give you like a very processed information. Okay, this is that, and this is that, and this is that. Sometimes it's good to kind of ask them, okay, what about this? Do you have any kind of quality control here? Anything that happened? Just to be aware. But then, once we have the FastQ, so the BCL, we move into the FastQ, and then I will tell exactly what is a FastQ file, and we do all the quality control. Anyone can tell me what is a FASTA file? It's a text file with sequences, right? Yeah. So it's something like that. It's kind of almost free kind of format, except that you have the sequence information, and usually you're going to start with this or with others, and then you have the sequence here. And that was the original kind of way to share sequences. And before the sequencing, the next generation sequencing, when you have thousands and thousands and thousands of reads, that was the way to share kind of information. I found by, using the second sequencing, this sequence that I think is about this gene in those samples. This is really important information to share because you collect here, 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 and then you start making. So they came out with this format. Now we have to, they require to extend it a bit more. Because we will have the sequence, but we will have something more. We want to know how certain are we about this A is an A, or this T is a T, right? So then we come into the FastQ format. How many of you have seen the FastQ format before? Okay, so still it makes sense that I give some explanation. So the FastQ format has to, again, have the sequence and the information of it, sequence name, whatever, information about the machine, for instance, where it comes, when, etc. Have the sequence and have some values here. This is actually the quality of the quality of how much we trust. This is a G, this is an A, this is a T. But encoded in a way that is very easy for ASCII in the ASCII code. So let's take a bit more into what is here. And I know this kind of, I don't want to go into the full detail, but those are important things because you will get public data. You don't load the public data. You can already get the process, or you can get the FastQ files, and then you say, well, I want to map it to the new genome kind of version HG20 because I was HG18. Fine. You need to really be aware of this. You really need to be aware of certain details. Why? First of all, this actually reflects the probability that the corresponding base call is incorrect. Imagine the DNA, or imagine the RNA, well, the DNA. It's always what we're doing. We have 4 nucleotides, so instead of saying the probability of this is a real T, let's say the probability that this is not a T, it's anything but a T. So if we do like that, like -10 of the log 10 of this probability, we get a score. The higher the score, the better, right? And this usually goes from 0 to 41, and the larger is the better. Why? Because if we have a quality of 2, means that it's quite likely that this is not, I mean, it may happen that this is not a real T, or is not a G, or we are not really sure what is that. The problem is the way to encode these, that are those numbers. We don't get the decimals, we go into the integers, 0 to 40, 41, into letters. Imagine this abecedary. We have different ways of doing that. The original Sanger, the Solexa, then Illumina was changing from time to time, and the last version is actually going back to the thread plus 33. I'm telling that because sometimes you go into the data, you go into the original FastQ files if they don't have this reprocess the quality. You need to check which codes they are using if it is not in the documentation. If it is said, okay, I use Illumina 1.3, fine, I know, this is thread 64. But if it doesn't say anything, you should be able to get some information from here. So the thing is, you compute this probability, but you want to compute this, you don't want to store the 0.0 blah 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 blah. I want to store something that gives me like, okay, this is good or bad in a way that I can easily store it. How can I easily store it? Using these ASCII codes, and then to different ASCII codes I'm assigning like a different value. And these values usually go from 0 to 41, and then you have here the mappings to each one of them. So I don't think we need to make a big kind of thing of that here, but be careful when you get data from the public repositories and to be sure that, okay, they are on the format that you are expecting. Okay, questions here? So now, and that maybe gives you some hints into that. Imagine I'm telling that I have for this sequence this quality, right? And this is actually not a letter, but it's a number that goes from 0 to 40. So I want to see, imagine if I have 1 million sequences. I want to see what is the distribution of quality over those million sequences in each location, right? So I take the 1 million sequences, I check this first location, and I check the quality. And I ask how good it is. And this is exactly what you do here. So you have from 1, imagine this is 1 to 100. So we are sequencing 100 per bases. And for the first one, we have an average of 30. And you can see that kind of the quality is very good without kind of variation for all the 1 million of them, right? Then it goes up a bit, it keeps going, and then after some time, 20, things are not getting that good. We have more variability, and in general, the quality is decreasing. This is what I was saying, that over the time you have more problems. What does it mean here is that you get some sequences that will go from 30, some sequences that go 34, maybe some more, but you have a distribution of them. This is very good to say, what is the quality of my samples? I will give you in a second a few more, more examples of things that are going wrong. That's I think it's a good question. For instance, I will show you, this is the FastQC pipeline, FastQC software, and they say like below 20 you get starting to get warnings. Sometimes if you get something like that, I don't know if you want to go to 30, and the thing is, if you get something like that, the degradation is at the end, this is what you are expecting. The other thing is you should look into every one of the samples, but you should look also into all the samples at the same time. Because if all the samples has this, then maybe you need to trim things, maybe you need to curate them, maybe you need to, I will talk into that in a second. But if one of them is going like that, this sample has a problem. You have several options like rejecting the data, or you can take out things that are not that good. Okay, but give me 1 second. I will work into that. Just this is if we look as a fragment, and the fragment has just one read. Yes, so we make this identification of fragment read. But it can happen that a fragment has two reads. That I was saying, you have a fragment. I'm sequencing in this direction, and I'm sequencing in this direction. So then you can have the quality for the both extremes, right? Usually, this is not the case, but some places I can see that the second read has a bit lower quality than the first read. And this is quite common because you first go all the process through the first reads, and then you start going with the other extremes. But I mean, in this case, and this is STATegra data, both kind of reads were quite good, and I was positively surprised. So what happens in a scenario like you are saying? What happens if it goes below 20? Much in this case, I will get really worried. This is the case that, for instance, I think in the FastQ show like a bad example. Okay. First of all, you were saying about what is the threshold. Usually you get good quality at the beginning. If you don't get good quality at the beginning, like 30 or more, I will kind of take out the sample, because that means that something has gone wrong. Usually you get good quality in the beginning, and then it goes down. So what happens here when it goes down? At some point you see that it goes down too much. It goes down below that. What that means, that you have a value here that maybe is a quality of 1. A quality of 1 is high probability of making a mistake. Maybe this is a G, but maybe this is a T. We don't know. So what are the options here? What do you think we can do? I mean, you can discard the full read, or you can say, well, anything, you check every one of the reads, and if the quality goes below that, you take out the end part. This is trimming, and you have like some tools here, and I included this paper on the Dropbox because it kind of compares different trimming kind of methodologies and approaches. Trimming is important, and I will show you why. There is more reasons for do the trimming, but it has a drawback. But we have to survive with it. I mean, the drawback is originally we have everything to be matching 100 per basis. Now we trim the data. We can trim in a hardcore way, saying like, I get only the 1 to 80, and everyone is going to be 80. But maybe you can want to trim based on saying, I only want to trim the low quality ones. So you will get things that has 60, something that has 80, something that has the entire 100 per basis, right? So you have different lengths. That's not a problem for the mapping, and usually should not be a problem unless it's associated to any kind of bias or any kind of association to that. So then there are different, I mean, this is about the quality, and one solution is to filter, trimming. I'm going to talk now a bit of some of the quality things that we need to be aware of. I don't want to go into full detail, but kind of give an overview. The next thing is the amount of CG content. So we have an expectation of what is the CG content that we are expecting in our samples, but sometimes, and then you compare what is the theoretical distribution one versus the GC content over the, sorry, not GC one, it's the same, but GC content distribution over the sequences. And then you compare them and say if they are too far apart. Two comments here. One of them is, of course, if we are thinking about DNA-seq, or if we are thinking about RNA-seq, the proportion of the GC content will be different. Because in genes, the proportion of GC content is not like the entire genome, right? So the proportion will differ, and we should be aware of that. Because some tools give you like a strong significance, this is not what we are expecting, but in reality, I mean, you need to take those things into consideration. The other thing is, imagine your sample has a huge GC content more than expected. And that can also be biological, because you have a set of genes highly expressed compared to others, and then it increases the proportion. From my point of view, what I think is more important is that all your samples, in your case, except if you have any reason for not, are having similar proportions. If you have one that is kind of completely different, and there is no biological reason for that to happen, then maybe there is some problems there. And this is one of the ways to identify the GC content problem. We were working with the data, and then there was always kind of some outliers there, and we were doing all the quality controls, and then we identified those samples that were behaving strangely, and then we were able to identify that they were behaving strangely, and they also had different proportion of CG content, and they were also associated to one specific library preparation. So it was like connecting dots and seeing like they are outliers exactly because of this reason, and because they were kind of really different from the proportion of the general sample. Not in the theoretical kind of explanation of the RNA-seq, more in the hands-on, I will tell you about, we will show what was the problems and how we try to solve this. There is a beautiful package called conditional quantile normalization that forces the distribution of the GC content and the distribution of expression over different lengths to be kind of similar. So for genes with the same GC content, you should have kind of similar distributions. But we will talk about more into the hands-on session. Another thing is k-mer overrepresentation. You can have a k-mer, it's like, I mean, we select 6 nucleotides, 8 nucleotides, and we are seeing how many times do they appear in the sequence, right? We can find these into the entire sequences that we are having, or we can also try to look them into different locations. And sometimes this FastQ, for instance, gives you like the which of them have been overrepresented in different locations. This is important because some of them can be the adapters from Illumina. We start sequencing, and we have like starting to sequence the adapter. Or because we have the fragments, and then we are starting on one end, and we go to the other, and then we are kind of sequencing on the other extreme. We arrive to the adapter. It's quite kind of common from my experience that you can end up having adapters in your sequencing. So what do? You do the trimming. You take out the adapters. So trimming tools right now are used for those two things: take out the low quality, and take out the adapters that you know are more probably part of the Illumina protocol. I think there is hard to say like what is the threshold. Usually what you do is to try to identify first. You rank the top ones, the most represented ones. Imagine here you have a lot of samples with this overrepresentation, right? Of the AAA. So imagine in this case how are those k-mers associated? Expected to are those k-mers in the protocol of the Illumina adapters, then you are not expecting to see them there. If they usually do also some kind of they also show it some kind of enrichment. Do you find any biological enrichment of those based on this sequence? Has been specifically anything on them? Not. If there is no extra information, we don't know anything, and probably we should do nothing. But if there is a specific information for that, like it's in the protocol, we should take it out. Again, you have a nucleotide. Instead of like the k-mers, you can try to say what is the percentage of the T, of the C in the different sequence on the different locations. We are expecting to have kind of a stable percentage, right? In many cases, what we see is that at the beginning you have kind of going up, down, and then it stabilizes. This is explanation from FastQ. This is expected when you have like random examiners, and then at the beginning it's not completely random what you get. But we should not take them out. Okay. It's a true technical bias. This is something which can be corrected by trimming, and in most cases doesn't seem to affect adversely the downstream analysis. Honestly, I haven't seen any clear answer onto those first ones, like why they are happening, yes. Why they are not affecting the downstream analysis, it's not completely clear to me. But in general, this is something that you observe here at the very beginning, and it's not a problem. The problem is it appears at the end. Because probably if it appears at the end, it can be some quality issues. Duplication. That's the other big general kind of comment. I was telling that we have like a fragment, and then we have the reads, and imagine we have the same read twice. Is this expected? Can be different reasons. One be that we have a PCR artifact, because doing the PCR, I mean, you get like this overrepresented too many times, and then compared to the rest, and then you have this overrepresented. You can have the cluster to be scanned twice. It's like sometimes if the clusters are coming really, really close by, or maybe it can be an issue separating them, or it identifies two and it's just one. And it tries to, I mean, there is ways to try to identify those things. But it also can be a biological duplication. So it's very important this, because imagine I'm getting a RNA-seq and I do 2 million reads. It's likely that I'm going to get the same sequence twice. In general, I mean, maybe. If I'm talking about the full genome as the DNA in 1 million reads, it's very unlikely that I get the same twice. So when we compare about DNA and RNA, it has very different expectations. So in DNA, usually we don't expect, if we talk about DNA-seq, that we are doing the full entire genome, we sequence the entire genome of an individual, we don't expect to have the same sequence twice, because we are going over the entire. If we go to many, many, many reads, maybe we can start expecting, okay, maybe I will get twice the same one, or 3 the same one. So I can set a limit. Maximum twice, maximum 3 times. If I go to exon sequencing, then it's a different situation, because exons is much smaller. I'm expecting to have duplicates. If I go to RNA-seq and I get like 100 million reads, like the ones we're having in STATegra, we are expecting to have a lot of duplications. In our case, we are getting like 97 in some cases duplication, but that's expected, because we are getting 100 million reads for something that is not that big. I'm saying that, and this is important. Originally, you can see papers that were saying, I do the RNA-seq, we filter the duplicates. You should take carefully, because there has been discussions about we do that, we do not do that. I don't do filter the duplicates myself. And in general, I think the trend nowadays is you don't filter duplicates. You try to identify, I mean, artifacts, etc. But in general, you expect duplication. This is a FastQ tool. There is also nowadays a tool, I mean, that can like a way to run it through R directly, and then you get the output from there. Myself, originally, what I did was to run it like command line, and then the output is on some file, so you know the structure, you can read it directly and import it. Another very important tool is PCAR, because PCAR gives you like a good, I mean, identifies like optical clusters for duplications, for all those things. Is very powerful also. It's more into the mapped files. I will talk into a second. But those are the tools for the, I mean, among them, the tools for checking the quality. So this part was about the quality controls. I mean, we have been talking about what is the NGS in general, generating the data, and then doing the first quality controls over the FastQ files. FastQ is it saves the sequence, and it saves the quality of how much we trust every nucleotide. And then we have certain actions here, and this is really important. I cannot say it enough, because we have, I mean, in our case, we have already tested that it makes a difference when you do the right timing, especially if you have some question marks over the quality of your data. Well, the next thing is, now we have some FastQ files that we have trimmed, and we think, okay, the quality is good enough, or, I mean, we can work with it. Our challenge now is to map to a reference genome. So, I will take it brief. There is a lot of papers and a lot of things to talk about those things, but we have a read, or we have two reads in a fragment, and we need to map them to the genome. So, if we kind of split into two things, one of them will say, okay, I want to map to the entire genome. And then you are using like BWA or bowtie, or there is like many other methods. Or I want to map to selected parts of the genome. Like I know I have a transcriptomic data, I want to map to the transcripts, I want to map to the genes. Then the problem becomes a bit more complex, because you have splicing junctions, right? And I will show you the problem exactly, but if you have a gene, and those are two exons, when you come to the transcript, you will see this. The read you will get can be on the splice. So when you back to the genome, you try to map this part, it will not fit, because it's one part is here, and one part is on the other side. So there are methods like Top Hat and other ones, many other ones, to try to cope with those situations. And I will show like few slides on that. But then, as I was saying, this is very data specific. So for RNA-seq, I mean, in general, we can use both methodologies. We use this one if we map to the entire genome, knowing that you have the splices. We can use this one if instead of mapping to the entire genome, you say, no, I have like my database of transcripts, and those are the possible transcripts, and I'm mapping to the transcripts. I will show an example later. What is the difference with the microRNA-seq? I mean, the reads will be kind of much shorter. That's one of the cases. So we will have the problem with the mapping and the quality of the mapping. We can decide that we want to go, but in the microRNA-seq, we don't have the problem with splicing. So somehow it's also kind of an easier part. Anybody can tell me what is the problem with the RRBS? Exactly. C-sensitive, exactly. And then you are not using the original reference, you're having one more, and then you have a problem with the mapping. So the usual methods are not kind of prepared for that, but you will talk about that later, and then you have software like Bismarck or things like that for mapping. So it has a challenge by itself. The DNA sec, the discussion will be on Wednesday. It's talking about how accessible is the chromatin. So if it is open or closed. So if it is open, we will get sequences there. And then, of course, we are mapping to the entire genome, and it will tell us information about where it's actually possible for a transcription factor to bind. So this originally easier. And for the single-cell RNA sec, we will go kind of the same situation for the mapping, same things. I think I will kind of stop this session here because that was the introduction to the full NGS. Then you will have the DNA methylation with specific pipelines. Then the DNAs with the specific pipelines. MacroRNA sec, we will talk about differential expression because basically it's the same pipeline, just with, I will not go into much details, I want more hands-on. And then the DNA sec, yes, you will have on Wednesday. So I want just to, this is the pipeline I'm going to show you in the RNA sec. I will just kind of change the slides, saying like things are growing also in the like single cell sequencing in many different aspects.